Yesterday we spoke about DNA sequencing as a technique and the gene variation in bacteria and antibiotic resistance and human variation. And today we will continue discussing with how the genes and their the mutations or the variants in them get exerted into different functions. Whether or not genes are functioning or not, there are several mechanisms of regulation, of which Thomas will present some. So the presenters today, the first one is Thomas Fleischer. He's a senior researcher and a group leader in the Norwegian Radium Hospital Institute for Cancer Research. He's an old collaborator of mine, and he took his PhD in my group, and he was a postdoc in my group, and has been successfully now launching a couple of projects on his own as a PI, and has been successful in generating money for his research, and has a couple of PhD students and master students, and hopefully a postdoc. Now, the next Thomas is a molecular biologist, both a web lab person and a computational biologist. The second speaker of this topic, of this day today, is Salim, who is a postdoc in the group of another young faculty, another senior researcher of the younger generation in my group, Xavier Tecpli, who was unfortunately struck with some very heavy flu and couldn't speak. So we had to either me take over his slides and present them for you, or what is much better, have his postdoc, Salim, who was so sporty that 15 minutes ago I called him and asked him, where are you? And can you present? He just defended his PhD thesis on a very, very, very interesting topic, which also concerns single cell analysis. So you will, yes, I think we should applaud Salim for whatever it was you were doing.
Exactly.
That was not that important, and you joined us, so we are very, very grateful that you saved the program. And you will see that he has a very, very fascinating story about how you can actually visualize and even model the behavior of single cancer cells. And then at the end, I will finish like I did yesterday, the last presentation, with one sum up of what kind of data we can acquire from tissues in a whole genome basis, both in the transcriptomics, but also at other levels. So with that, and saying that again, we are very thankful to be part of this course. And yesterday there was very nice discussion. I hope today the same. We could start with you, Thomas, maybe.
Thanks. Yeah, it is exciting to be part of this course. Fun to be teaching. I guess I can share my slides.
Yesterday everything was okay with this.
Yeah.
Let's see.
So
Yes,
do you
we
now
see.
see my full screen slides?
Yes.
Yeah.
Very
Cool.
good. I will switch off microphone not to load the line. Okay?
Do you also see my pointer? This platform is.
Yes.
Yeah. Great. Thanks.
Works well.
Yes, so Vesla already said some things about me. I've been working on epigenetics and DNA methylation since 2010, when I started my PhD in Vesla's group. And that's what will be the focus of today's talk. Epigenetics in general, I'll describe that to you. I unfortunately don't know exactly what you know about epigenetics from before, but I will go relatively basic for at least the first half of the talk. Feel free. I know that the Zoom-like platform is not the best for discussion on the way, but do feel free to interrupt me as I go, or we can take the discussion at the end. This is up to you. I don't mind being interrupted. So the title, Control Command of Gene Regulation, this is, I guess, a statement to be debated, which, yeah, we'll touch on. So the outline is that I will introduce epigenetics with a certain focus on DNA methylation, but also on the other aspects of epigenetics. As I am a cancer biologist, the presentation or the lecture will be colored by cancer, even though a lot of these things are common to all human states, including diseases. But I will mention some of the samples that we analyze. I will tell you quite a bit about how we measure DNA methylation, and I'll also include single-cell methods, because that is a very important and exciting new phase in the whole cancer research, our availability of the single-cell methods. And I will also tell you more about epigenetics in cancer, and specifically a little bit about breast cancer and some examples of research in my group. So the motivation for studying epigenetics, I think, is intriguing. So all cells in our bodies have identical genomes with certain exceptions, but still the cells behave extremely different. You have eye cells, you have skin cells, you have liver cells, just as examples. Even within the eye, the myriad of different cells behave very differently and have different jobs. And I would argue that the differences between cells in our bodies is much greater than the variation between us. We are quite similar to each other, actually. So all these differences are caused by stable differences in gene expression, that is gene regulation, that is actually causing this tissue differentiation and tissue identity. And it is clear that epigenetics is thought to be the major player in the regulation of stable regulation of gene expression, as well as and consequently tissue differentiation. And what's intriguing in a cancer setting is that cancer cells are essentially cells that have lost their identity. They don't follow the rules that are defined by the tissue they should belong to. And that makes it appealing to think that epigenetics plays a role. The most basic epigenetic mark or alteration is the DNA methylation. It's the addition of a CH3 group to cytosins. And this is found where cytosins are followed by guanines, and we call these CpGs, CG dinucleotides. The cytosine looks like this. The addition of a CH3 molecule is catalyzed by DNMT enzymes. And on the DNA strand, it looks like this. This is an unmethylated CpG with no addition, and a methylated CpG with a CH3 group added to the C. And you will from time to time see these gray boxes being an explanation of the abbreviations. CpGs in general are underrepresented in the genome because they are susceptible to mutations. But there are regions in the genome with high CpG density, meaning basically that there are many CG nucleotides in certain regions, and these are called CpG islands. And here you can see just from the genome browser, there's an example of a CpG island with lots of CpGs, and these are often located in the promoters of genes. And here you see a CpG island that is located in the overlapping promoter of these two genes, BCAP31 and ABCD1 is just an example. But this is very often the case that you see the CpG islands in the promoters of genes. And in normal tissue, most of these CpGs are unmethylated, here denoted by just Cs. And this allows the transcriptional activation of genes, while CpGs outside the CpG islands are often methylated in a healthy genome. While in cancer, we see a certain number of genes, especially tumor suppressor genes, they get hypermethylation or methylation of the CpG island. And this has been known to cause transcriptional inactivation of, for instance, tumor suppressor genes. Here's really a rather similar view or depiction of the DNA methylation and the genome, but here you see both of these genes on the same genome. Now here it's the same DNA strand. This is an active gene, this is an inactive gene with the promoter, but here we also include an enhancer and a transcription factor. So if we focus on the vertebrate genome, we see there where there's high bars of gray, it means that the DNA is methylated. Well, if there's a low white, it means that the gene is unmethylated. So what we see is that in general, when there is an active gene, the enhancer is also unmethylated, allowing transcription factors to bind, or at least there's a thought that there's a connection between the epigenetic state, the DNA methylation of enhancers, and the promoters and the activity of the gene. While in inactive gene, the enhancer will be methylated and the promoter will be methylated in many cases. But this interaction between transcription factors and the DNA is rather complicated. Here's 5 examples of how one envisions this interaction to occur. This could be either that there's a transcription factor that don't care about the methylation state, and it can even remove the methylation once it binds. There might be a transcription factor that binds specifically to methylated DNA, or there might be transcription factors that are sensitive to methylation and are inhibited from binding when the DNA is methylated. One can also know about these MBDs, which are methyl CpG binding domain proteins. Heavily methylated DNA can recruit these proteins, and these can cause steric hindrance of binding of transcription factors. Then the final one is sort of connected with the first one. Pioneer factor is a transcription factor that can bind the DNA and remove DNA methylation, open up the chromatin, and that way recruit more methylation-sensitive transcription factors. DNA methylation can be read, it can be written, and it can be erased. So if we start with a methylated CpG, this will generally be kept methylated by the replicative maintenance performed by DNMT. So when the cell divides, methylated DNA will stay methylated by this enzyme. But one can also see a passive loss where this enzyme is not active or is not present or is not present in high enough concentrations, leading to loss of methylation. And there's de novo methylation, which means the methylation from scratch, being catalyzed by these 3 enzymes, DNMT3. There's also active demethylation catalyzed by the TET enzymes. And this leads to hydroxymethylation, formal cytosine methylation, and carboxyl cytosine modifications to the C, that through these 3 mechanisms, the CpG becomes unmethylated. So this is the writing and erasing part. The reading part we have already been mentioned on the last slide. In addition, there are CXXC domain proteins, such as these, that will actively bind to unmethylated CpGs, and they can cause recruiting of other proteins. And the same with the MBD molecules that can either be recruited and most, as I said, cause steric hindrance for other proteins, but it can also recruit other proteins. So this is a complex mechanisms that is not fully known yet, but these are at least ways of reading the DNA methylation signal. Some parts on histone modifications as well, being an important part of the epigenetic code. So this is a histone. The DNA is coiled around the histones, and the tails, the peptide tails that protrude or extrude from these histones, they can be modified chemically with chemical groups that have different properties. And this includes acetylation, citrullination, methylation, and phosphorylation, and ubiquitination. And these, again, have different properties, both also when it comes to positive and negative charge. And again, here, there's these are dynamic marks, so they can be written and they can be erased. And they can be also be read by molecules that recognize the chemical groups and recruit other proteins so that you, for instance, will loosen up or cause transcription of nearby genes. So in order to try to get an overview of what epigenetics is and why and what it all boils down to, epigenetics are modifications to the DNA molecule that does not change and doesn't involve changes in the base sequence. And through these mechanisms, the organism can turn genes on and off. So we have been through DNA methylation, histone tail modifications, histone variants I haven't actually mentioned, but these are, the histones are composed of a protein. It's actually a protein complex, so you can switch out certain parts of this protein complex, getting new properties. Nucleosome positioning, I didn't mention that, but the nucleosome is just a name for the DNA and the protein together. And this can shift around, so you can move these histones around, sort of coiling it along the DNA. And lastly, and importantly, is the chromatin looping, which is basically the chromatin loops, and you can get contact points between DNA that is far away from each other on a genomic view. So what you see here is an inactive promoter and an inactive enhancer. We see repressive histone marks, we see demethylated DNA, and we see that the histones are placed close together. And we see the same thing in the distal enhancer being located far away from this promoter. This is the same stretch of DNA, but in an active state. So here we have active histone marks. We have lost DNA methylation. There's more distance between the nucleosomes. We have recruitment of transcription factors to the enhancer. We have recruited even more proteins, mediator complex here, and we have recruited RNA polymerase II, which is the direct player in transcribing the DNA into RNA. So if you put it simply, epigenetics can be viewed as just whether the DNA is available for proteins to bind, whether it's open, available, or if it's closed, compacted, not available for proteins, for instance, polymerase II, to transcribe the DNA into RNA. So that was it for the introduction to epigenetics. Now, these are some of the samples that we very often use. There's others, so just as simplification. We very often use immortalized cancer cell lines. So these are probably at some point taken from a patient, but they've gone through certain steps to be able to grow in a dish. And this allows us to interfere with the cell lines and do experiments on it. Tumor biopsies are, for instance, in our case, taken from the breast. You operate out the tumor from the breast, and then we get it in our department. We take it in a dish and we cut it up and have it like this. Or it can be blood samples or other disease biopsies. So once we have the tissues, whether it be cells directly from a dish or tumor pieces, we isolate DNA and RNA from these samples. So this can be done in very many ways. It's been, of course, done for a very long time, but we use in general a method that is column-based, meaning that the DNA will attach to a column, and then you can elute this DNA. But the clever thing in this approach is that the RNA will then be allowed to stay in the flow through, and you can add another column that will attach the RNA. So we basically get RNA and DNA from and even total protein. We get this from the same sample, and this is done using a robot, so it's relatively little hands-on. A very practical way of isolating DNA and RNA. So this is a QIAGEN kit and QIAGEN instruments. And of course, for the DNA methylation, what we're interested in is the genomic DNA. This is a slide taken from this new paper. It's very busy, but it illustrates that DNA methylation can be measured in very many ways. Don't spend time looking too much at this. If you're interested, you can go and look at the paper. I'll, of course, share the slides if that's something you want. But I will try to focus on the three different methods for principles for measuring DNA methylation. So one is the first and perhaps the most common is the Bisolfit conversion. So what this method does is that by treating with this bisulfite reagent, you convert unmethylated cytosines into uracils. And inevitably you will do PCR or something, and then you will transform or convert the U further into T. While the methylated CpGs are protected from this conversion. So what you have done now is that you have altered the epigenetic mark of DNA methylation into a sequence polymorphisms. So now you can actually analyze these DNA in the same way you would sequence your DNA for mutations or other things. The problem with this method is that it reduces the complexity of the DNA, meaning that you have to sequence a lot deeper to be able to align properly. And in addition, the bisulfite conversion is quite tough on the DNA, so we will fragment the DNA, and it will also degrade a lot of the DNA, meaning that a lot of the sequencing methods that you would use on regular genomic DNA is hard to use on bisulfite converted DNA. The other method is using methylation-specific restriction enzymes. This method cuts the DNA where it's not methylated, and you're able to remove the cut DNA from the analysis and only sequencing what's not cut by the restriction enzymes. An affinity-based methods uses some sort of proteins, antibodies for instance, that bind to the methylated DNA and allows you to pull down and sequence. So I'll go through some of the most important methods, at least for cancer research. The methods will be a little bit different depending on what sort of applications you want, but I think these methods are what's generally used quite a lot in research. So the Illumina Infinio microarrays have been extremely popular in the cancer research because they we're able to analyze very many samples, which is important. So this method, again, because we have now, it's based on the bisulfite treated DNA, meaning that we have now converted the DNA methylation signal into a sequence difference. And the only thing that happens is that the probe will hybridize to the DNA, and depending on whether the CpG has been converted to T or not, there will be a different nucleotide with a different color that will attach. And this color can be read out in a scanner. This will return for every CpG that is probed. It will return a value that represents the percentage of DNA molecules that have a methylated C at this loci that the probe interrogates. So it's well suited for high throughput, and it's a very streamlined protocol. The limitations is that there's a limited number of CpGs. We see the currently used technologies using either 450,000 or really the current technology is 850,000 CpGs. But this is still a very limited number considering there's 32 million CpGs in the genome. And these CpGs are pre-selected, so you can't, in theory, you can't identify something completely novel. It requires relatively good quality of DNA, but that's actually a requirement of all the methods. And it cannot detect hydroxymethylation, which is part of active demethylation. Incomplete conversion may result in false positive, so if the bisulfite conversion is not complete, but this is also true for all the other bisulfite methods, and it is becoming less and less of a problem. I think this bisulfite conversion is now very efficient. Then there's sequencing, also using the bisulfite sequencing. It's either whole genome bisulfite sequencing or RRBS. The whole genome, as it says, interrogates the whole genome, approximately 32 million CpGs. RRBS uses a restriction enzyme to actually cut away regions that are not, that have a small or that are CpG poor. And in that way, being able to enrich for certain regions in the genome with a lot of CpGs, and that way allowing it to sequence slightly less than if you just do random fragmentation, having to sequence everything. But after that, the library preparation and so on is the same. You do bisulfite conversion afterwards, and you do deep sequencing. The limitations here, it's still in 2021, this is more expensive than the microarrays. There's more complicated bioinformatic analysis. The RRBS is called reduced representation bisulfite sequencing, by the way. It covers approximately 2-3 million CpGs. So here the limitation is that you actually lose the regions that are poor in CpGs, and this might still be important regulatory regions. So the final method slide, I think, is MRE is a methylation sensitive restriction enzyme based. So here you use these enzymes to cut the methylated regions, and you're able to collect the methylated CpGs, and you just sequence that, and you get peaks, just like in ChIP-seq or something. You get peaks in your DNA, on your reads where there is methylated CpGs. Sort of similar on the MEDIP-seq is methylated DNA immunoprecipitation using an antibody. The same with the methyl binding domain protein seq. There's also these proteins bind and are able to collect the methylated CpG. So the advantage here is that you cover a lot more of the genome compared to both the arrays and the RRBS. The main limitation is that it is less accurate, and it will especially be less accurate where there's few CpGs and relatively little methylation. And you don't get these percentages of DNA methylation that can very often be very useful. So an overview of these methods is that the whole genome bisulfite sequencing, yeah, you have been... You have the genome coverage on the x-axis and the throughput on the y-axis. So the whole genome bisulfite sequencing has Can Anton please mute? Yeah, and the MEDIP and the MBD-seq has higher coverage, but lower coverage, but still higher coverage than these. All the Illumina DNA beads chips, as we talked about, has a higher sample throughput. Now, I would argue that all of these methods are actually too low. You're able to sequence more than one sample with the whole genome bisulfite sequencing, and more than 200 or more than 100 with the Illumina. And even the RRBS was just published 2,000 samples from the Metabrick breast cancer cohort publicly available. So even everything here moves up really in today's research field, but the shape of these methods are still the same. So the choice of method will depend on many things. So of course, the number of samples that you have and the money that you have available. But I guess also in practice, the previous experience, what you're happy with, what you have experience in the lab, what you have experience in your institute will also depend, because realistically all these methods are performing quite well. So now I'll touch on single cell methods, and first a little bit of motivation for why we care about this. So here you see 3 examples of any biopsy really, but let's call it a tumor biopsy. And then on the left, you have a completely homogeneous tumor where all cells are the same. In the middle, you have a very heterogeneous tumors with very many types of cell. And on the right, you have a tumor where a lot of the samples are, cells are similar, but there's some rare subpopulations. So now we can look at this as populations, not really as single cells, I guess. So in cancer, this picture may be the case both for actual tumor cells that are different, but also importantly, these populations might represent non-tumor cells, such as immune infiltration, fibroblast infiltration, endothelial cells. So this is really a very complex picture of different subpopulation of cancer cells and infiltrating cells. And using a bulk method, bulk now means that you just take a whole biopsy or a whole pellet of cells or whatever. And using this, in this case, because if you average it becomes the same color red, using a bulk method, you would not be able to detect that these three tumors are different. And you would certainly not be able to tell anything about the subpopulation, even if you knew they were there. But using single cell methods, we're actually sequencing each individual cell. And this allows us to say that, okay, in this tumor, they're all actually the same. Well, we can detect the properties of all the subpopulations. And in this case, we're able to maybe potentially detect the characteristics of this one rare subpopulation. And it is so that, I mean, I don't know how much you know about cancer biology, but these hallmarks of cancer are these different things that a cancer or a disease needs to be able to do, such as being able to replicate indefinitely, avoid the immune system, being able to get enough nutrients, alter the metabolism, being able to invade adjacent tissue. All these things are essential for the tumor. But it might not be that every cell in the tumor has the capability of doing all these things. So that's where I think this intriguing concept of division of labor. So basically the different subpopulations of tumors are in the tumor may be able to actually do the different things, while not all of the populations are able to do all things. And this illustrates how important it is for us to be able to interrogate each individual subpopulation. So here are some examples of methods. You have the bulk methods in the left column. So RNA-seq, microarrays for gene expression. For DNA accessibility, you have ATAC-seq, histone modifications, ChIP-seq, for instance. DNA methylation, we have been through it. Bisolfite sequencing, RRBS. They don't mention the arrays, but they're here. And the single cell methods that can do the same thing. So there are many single cell methods. Single cell RNA-seq, single cell ATAC-seq, but as you see, single cell ChIP-seq, single cell cut, there's many. But the thing is that most of these methods are not, at least for cancer research, they are very difficult to use, either because they require much more material that we have, or they're extremely expensive to do, or they're only, I mean, for instance, a single cell bisulfite sequencing has only been shown to be able, there's only a couple of labs in the whole world that's able to do them. So that's why I say here that not all of these methods are actually in routine use, but perhaps the two most important and the two methods that have produced the most results is the single cell RNA-seq and the single cell ATAC-seq. So the single cell RNA-seq you will hear a lot more about from Salim in the next lecture. So I'll touch on the single cell ATAC-seq because I think some really interesting results will come out of that. So the ATAC-seq is based on cutting with this, anyway, cutting the open available chromatin and identifying the fragments that were in the accessible regions. So then just regular library construction and sequencing. There's of course a lot more complicated biology going on and technology going on here, but that's the idea. So in a single cell approach, there is also different ways of doing that. But one way is this bar-coded gels and you have the nuclei. And in an oil, you're able to attach single nuclei to these bar-coded gel beads. You collect them and you're able to actually sequence each individual nuclei. So in that sense, it really opens up for new exciting insights into intratumor heterogeneity, be that the epigenetic properties of specific cell populations, as I mentioned on the last slide, or even very importantly in treatment resistance. Let's say you have tumor that you have the biopsy before treatment and you have a biopsy after treatment, and you're able to actually see which properties have arisen in the resistant cells that are left after treatment. And this will, I think, bring a real new insight into the cancer research. I'm not sure about the time. I don't remember when I started. Does someone remember?
Something like quarter past 2, I think.
Oh yeah, great. Then yeah. So this figure from this publication here sort of illustrates very simply the vision of omics research, and probably something that Vesla also touched on, where we have a lot of cancer patients, mostly cancer patients, but we may also have healthy controls. We collect a lot of molecular data. We collect the epigenome, the transcriptome, genome, proteome, and metabolome, for instance. And our aim is to identify a causal contribution of all these different pieces of a puzzle, and by that identifying specific disease-associated genes or alterations. And this can be used both to diagnose patients better, to say something about the prognosis, as well as which therapy they should receive. And as a part of that is patient stratification. And I would argue that this arrow could actually come from all levels. You don't need to understand the biology, even though that's an advantage, in order to perform patient stratification. And that way, you can, when you have patient stratification, you can contribute to tailored therapy, giving different therapies to different patients. Cancer epigenetics is really very, very complicated. There's thousands and tens of thousands of papers describing cancer epigenetics. So I actually think this figure from 2014 sort of illustrates a lot. The most important thing to remember is that realistically, what all these epigenetic alterations do is affect tumor suppressor genes by inactivating them or by activating oncogenes. And both of these things will obviously contribute to progression of tumors or even chemoresistance. This is just an example from ovarian cancer, but it's the same for all cancers. So this might be hypermethylation, increased methylation, loss of methylation. It may be condensation of the DNA through certain histone alterations, and the opposite with the condensation of the chromatin. And also our microRNAs are sometimes included as epigenetic marks, even though I'm not included it in my general talk. But it's easy to see that these microRNAs can also affect tumor suppressor inactivation and increase oncogene activation. That said, I have some interesting observations that I thought I could mention. Mutations in genes coding for epigenetic factors have been shown to play an important part in certain cancers. And by getting mutations in these factors, you can impact epigenetic homeostasis and potentially leading to tumorigenesis through altered epigenome. But in this case, it will start with mutations in the epigenetic factors. There's also decreased activity of chromatin modeling enzymes, promoting global chromatin restriction has been observed in many cancers, as well as promoter hypermethylation that suppress tumor suppressor genes. So these are things that are in general observed quite often, but it's certainly not the only effects that can be seen from in cancer. So another interesting observation I find is that, again, the hallmarks of human cancer can potentially be achieved purely through epigenetic deregulation. And here in this publication, they suggest this concept of pure epigenetic alteration. Of course, this is speculation, and how often this happens is they only speculate, but I think it illustrates the point that it doesn't necessarily, cancer doesn't necessarily need genetic alterations. And I think understanding of the epigenome alterations in cancer is still, even though we have studied it for a long time, it's still in early stages. And I think, again, that single-cell profiling will be very important. Another thing I haven't mentioned is ex vivo, advanced ex vivo models and in vivo models. I think this will really bring the research forward. So these are, instead of using cell lines, you take samples from a patient, and this can now include the microenvironment. It can include immune infiltration, fibroblasts around, and it can even even include physical a grid and a 3D structure. And these models, in vivo being mice models, for instance, or models in mice, no, in animals, and these models give we're able to manipulate cancers in a completely different way. And I think this, together with single-cell profiling, at least in my view, is two of the most exciting new avenues in cancer research. So then I'll have close with a little bit on breast cancer. The breast is composed of this, is mainly composed of adipose tissue, but there are lobules producing milk that and ducts where the milk is transported to the nipple. A cross-section of a duct shows that there's epithelial cells and myoepithelial cells, and these cells are where the cancer arises. And the cancer starts by rapidly dividing, and after a while it's able to breach this membrane that is around the duct and invade adjacent tissue. And there's genetic and epigenetic alterations that increase in this progression of cancer. Estrogen receptor is a very important part of breast cancer. Estrogen receptor is a receptor and a transcription factor that's when it binds to estrogen, it will go into the genome and activate other genes downstream, estrogen targets. So what happens in estrogen receptor positive breast cancer is that this receptor is overexpressed. So there's much more estrogen receptor than it's supposed to be, and that again activates a lot of downstream genes. And that causes proliferation. Similar, HER2 status is an overexpression of this surface receptor, also causing increased proliferation. Both these receptors have drug targets. Also, we can divide breast tumors into molecular subtypes, luminal A and luminal B being ER positive, HER2 enriched being HER2 positive, and basal-like being negative for these two receptors. Luminal A is generally have a low proliferation and good prognosis, while basal-like and luminal B, HER2 rich, are all with moderate to bad prognosis. What we identified in 2017 was that DNA methylation is very different between ER-positive and ER-negative tumors. So what you see here is a heat map with patients on the x-axis and CPGs, the CPGs that we identified on the y-axis. And you can see here that the ER-negative ones, are on one branch in the dendrogram, while ER-positive with a lot lower DNA methylation, the blue, than the ER-negative. And what's important is that these CPGs that we identified, they were very, very often found in estrogen receptor binding sites, because we know where the transcription factor binds. They were found in FOXA1 binding sites and GATA3 binding sites, all transcription factors that are known to be very important for breast cancer. So what we then saw was that the methylation was lost in the ER-positive breast cancer compared to both healthy breast and the ER-negative breast tumors. So this allowed us to think that the loss of methylation may allow transcription factors to bind and increase the expression of estrogen-related genes. And here's an example, because not only were they in enhancers and transcription factor binding sites, these CPGs were also often found in loops between, so this is an example of a CPG found in an enhancer binding site of ER and FOXA1 and looped to the start of a known gene to be part of estrogen signaling, to be a target of estrogen. And RAR alpha is this example, and you see here a correlation between the DNA methylation and the gene expression of this gene. So what we think is that there's a loop here with enhancers recruiting FOXA1 and estrogen receptor, consequently recruiting polymerase II, expressing the gene in the ER-positive tumors. Another thing, and the reason why I mentioned the luminal A subtype, is that using DNA methylation, we're able to actually subdivide this relatively homogeneous patient group into two groups, one with a lot better prognosis than the other. And this shows that DNA methylation may increase our understanding of not understanding, but increase our resolution when it comes to subclassification of breast cancer and actually help treatment decisions for patients. So that was it. I think not very much, gone over time. I would like to give this summary. Epigenetics play a major role in regulation of tissue differentiation, and it affects expression of genes and how proteins binds to DNA. Single cell technologies are really opening up for new knowledge of tumor heterogeneity, as well as cancer-specific alterations that happen in certain populations. Epigenetics is drastically altered in cancer and may contribute to carcinogenesis, and a lot is still unknown concerning really the disease-driving effects of the alterations. Breast cancer is heterogeneous. It is a very different disease with a lot of heterogeneity and novel biomarkers to classifications. The patients can be useful. And finally, that we saw that enhancer methylation is important for transcription factor activity and the development of ER-positive breast cancer. Well, thanks a lot for your attention. I hope you found it interesting.
Yes, Thomas, thank you very much. If somebody has questions, it was amazing presentation, a little bit obscure and mysterious for my knowledge. But still, if somebody has specific question, because I have general question which I still don't understand, it's interesting that I understood applied part better than the beginning when you made your introduction, and I still don't understand if regulation and all these events come from a genetic to epigenetic levels, but all epigenetic molecules which participate in epigenetic events, all these proteins which associated with enhancer, trigger, all involved genes, etc., they are also encoded by their own genes, and to regulate something, to make differences in population of cells, whatever. It can be tumor cells, it can be, I don't know, maturation of something in differentiation. It's necessary some initial trigger, because we just go to another bridge of this ship, but I still don't understand who. Maybe they are known, for example, for cancer mutation and molecular events which happened not directly in the tumor or proliferation controlling genes or tumor suppressor genes, but in genes which are responsible for such things like methylation, demethylation, forming of this complex of enhancer and how it's called, promoter, etc. Can you try, or maybe, I don't know, someone can explain this in other words?
Mm.
I understand what I mean, yes?
Yes, I understand what you mean. So first of all, I think what you're asking is, of course, very pertinent, but I think simply we don't know the complete answer to your question. So you're asking sort of what comes first, what are...
The egg, chicken.
Yes, and so what I can say from this is something that interests us, and what I can say, for instance, we were very excited to see that it was such a clear signal with the loss of methylation at the enhancers, being associated with high activity of transcription factors. But we couldn't say from this observation whether it was some activating mutation in a transcription factor that increased the activity of the transcription factor, which then removed the methylation, or if it was some methylation. It could also be the origin, the cell of origin being unmethylated at the enhancers. So we don't know the answers to that yet, but what we can say is that there's no simple mutation that we can identify that explains why we see an altered epigenome in breast cancer, for instance. So this is basically, and what I also can say is that we are working with CRISPR experiments where we want to do CRISPR epigenetic editing. So where we want to go in and see if we make specific alterations, how is the causal contribution of that? That's one avenue. Another avenue is to knock down transcription factors and see if these transcription factors are not occurring in the cell. Will this alter the epigenome? So all these things are just small pieces of the big puzzle that you ask.
Exactly.
Yeah, that's, I think, the answer I can give.
Yes,
Do you have anything to...
I'm satisfied. Yes, absolutely. If somebody has other more specific questions than my one.
Hello. Thanks for your presentation. Very large topic. I have a very basic question. Do you know of any crosstalk between DNA methylation and histone modifications? Can somehow the DNA methylation affect the chromatin state, or could specific histone modifications predispose for higher DNA methylation?
Yeah, so functionally, I'm not currently aware of that, but I'm pretty sure that there will be literature on that, because there's done a lot of sort of basic research into epigenetic mechanisms. Although the problem is that a lot of the organisms that are used, the model organisms that are used for this research, don't have DNA methylation. So that's a limitation. So a lot of it is done in yeast, for instance, and there's no DNA methylation there. But I think, so that's on the mechanistic part. That I can't answer. But on a non-mechanistic view, of course, there is a high overlap between repressive histone marks and DNA methylation. Where there is repressive marks, there will also very often be high DNA methylation. But again, what the mechanisms of recruiting of the enzymes there, I don't know.
So can we consider the DNA methylation as evolutionary New Year if we don't have it in yeast? Okay. Yeah. Okay. Thank you.
Thank you, Emil. I think we should go ahead because we're a little bit out of frame, and if there will be time and interest, we can discuss in the end all raised questions. Okay, so Christina, what happens? I don't know. Visela, sorry, I'm sorry. I'm very sorry. Visela, you will present another Dr. Salim. You already did something, but please, introduction in 1-2 sentences. Microphone is switched out. Microphone, Chris.
Yes. Sorry, I was just because people are coming in and out all the time, so I wanted to spare you. So the speaker we had planned initially, Xavier Tekpli, got very sick, so he will hopefully give us his story in the next seminar. But we have Dr. Genome Salim, who is a doctor just a few weeks now and who had a fantastic dissertation defense and who has also been working on single cells. So he would be telling his story or his studies on single cell analyses, which Thomas already introduced to us. So why are they important and how can we study them? Salim, thank you again so much for stepping into this in 15 minutes. And I'm very much looking forward to your presentation because I know it's very good.
Thank you. Okay, I will share my screen.
Mm-hmm.
Do you see my screen now?
Yes.
Yes. Mm-hmm.
Okay, so hello everyone. The title of my talk today is Towards a Computational Rooted Approach to Cancer Diagnosis and Treatment. I will be showing today how combined computational experimental approach can help uncovering the secrets of cancer leading to better therapies. I will be showing some of my PhD work. When multiple cellular life began, so did cancer. Several pieces of evidence strongly indicate that prehistoric animals such as dinosaurs had cancer. Cancer is a disease of multicellularity. It has been reported across the tree of life from Hydra to human. CT scans of Egyptian mummies reveal oldest known cases of breast cancer and multiple myeloma, which is a type of bone marrow cancer. Why cancer is called so? The word cancer came from Hippocrates, the father of medicine. Hippocrates used the words carcinos and carcinoma to describe tumors. These two terms usually were used to describe a crab, which Hippocrates thought a tumor resembled because of the finger-like spreading projections from the tumor. But nowadays, what do people think the cancer is? Maybe patients will say it's a terrifying disease. Scientists for sure see it as uncontrolled development of cells. From medical perspective, cancer is a very heterogeneous group of diseases and the emperor of all maladies. Cancer is a disease in which abnormal or transformed cells grow and multiply when they shouldn't. These cells no longer listen to the body cues to stop dividing, no longer listen when the body says it's time to die, no longer listen when the body says it's time to stay in place, but they want to travel to distant places in the body to form new tumors. This process is known as metastasis. Today, cancer is seen as a disease that affects both genders roughly equally, and the risk of many cancers is highly associated with age. A few years ago, a nice article appeared in Times magazine describing cancer in very descriptive way. So they called cancer an intricate and potentially lethal collaboration of genes gone array, of growth inhibitors gone missing, of hormones and epigenomes changing and rock cells breaking free. It works as one great armed force. Metastasis is the leading cause of breast cancer associated death. The metastatic journey usually starts with single cells penetrating the wall of blood vessels, entering the circulation in a process called intravasation. These circulating cancer cells are potential metastatic seeds that might succeed in initiating secondary tumors. But what migratory characteristics of speed and persistence these cells must have to make a bigger tumor with more intravasation? This brings me to the first aim of my project, which is finding out the impact of cell migration on both tumor growth and intravasation in breast cancer. To address this question, we studied the migratory movement of several breast cancer cells using several migration assays by letting breast cancer cells migrate randomly and through wounds. And here there is an example of triple negative breast cancer cells. Some of these cells are expressing GFP, localizes to the nucleus, and these cells are migrating randomly in dense population. And the purpose of mixing GFP cells with the regular cells for optimal tracking efficiency. After collecting these images resulted from the live cell imaging, we struggled finding optimal tools to track these super migratory and super proliferating cells. So we decided to make our own tools, and these tools now available freely at the GitHub repo of Oslo University. The first tool is called CellMap Tracer, which is a multi-platform tracking tool for long-term migratory and proliferating cells. It's written in MATLAB, but it does not require any coding skills because it's available with the user-friendly graphical interface. The process starts by loading the TIFF files of the live imaging, and then the user will select the cells to be tracked in semi-automated fashion. And also they can monitor the cells during the tracking and also correct if it's needed. And here it comes the power of this software, because usually the algorithms do a lot of things that we can't see. And in the end, maybe or maybe not, can these results be trusted. But here you can see everything, and if you feel that the software did something wrong, it can be corrected. Users can plot the tracks of the cells and their daughter cells in multi-generation plots, and also the software provides lineage trees of the cells and their daughter. And by looking at the trajectory time of the cells that had complete cell cycle, we can estimate the doubling time in real time of these cells. And also we can also study the synchronization and the heterogeneity between the daughter cells from different points of view, from migration perspectives and cell division perspectives. On top of that, cell map tracer is associated with Fuji systems, fluorescent ubiquitination cell cycle-based indicators. It's a straightforward process. The user needs just to load the TIFF files, the RGB files, and the tracking data. And then the software by itself, it will analyze the signal throughout the tracking course. And that will help us understand the dynamics of the persistence and the speed across the cell cycle phases. And as you can see here, we found in RPE cells and breast cancer cells that cells during the S phase are significantly slower than G1 and G2 phase. When we looked at the persistence, we saw that these cells seem to have almost same persistence during G1 and S phase, but they increase their persistence during the G2 phase. The software can help us also find behavioral characteristics of the cells. So while we were monitoring the cells, we noticed that the cells just before generating two daughter cells, they have a kind of a big jump. We call this jump the speed terminal jump, and it's found in about 65% of the cells. This paper was published in Cells a few months ago. The other tool was named Cell Migration. It's an integrated R package for tracking single cells and analyzing their trajectories. It was published in Bioconductor, which is a container for well-documented packages. It enables users to track cells and study their trajectories at single cell level, as you can see here in the upper panel, and also at populational level, using several sets of measurements for persistence, directionality, displacement, and speed. I'm not going into details now about that because we have more than 40 measures, so I will be focusing only on two terms, persistent and speed. Persistent is the ability of the cell to maintain one direction during the movement. And here we have two examples of two tracks. The upper one shows a cell that is highly persistent due to the straightness in the track, whereas the lower one is for a cell with low persistence because of the zigzag in the track. There are several ways to estimate the persistence, and one of them is called the persistent ratio, where we just divide the number of persistent steps by the total number of steps. But what is a persistent step? How could we know if a step is persistent or not? We look at the angle corresponding to the step, and if that angle is less than 90 degrees, then we call it persistent step. If it's greater than 90 degrees, then the step is not persistent. And in this case, the cell has high persistence. Usually the persistent number values will be between 0 and 1. 1 refers to very persistent cell. We studied the persistent ratio of thousands of breast cancer cells to see the ranges of the persistent values of triple negative aggressive cells. The other term is speed, and it's obvious how fast or slow the cell is. And also we studied the speed of several thousand of breast cancer cells. And we used these measures to make a matrix of speed and persistence based on the ranges of speed and persistence the cells use. So here, blue numbers refer to persistence and the yellow numbers refer to speed. And each combination is used later on in the modeling. So here we have about 130 different combinations of speed and persistence. To predict the impact of cell speed and persistence on tumor growth and intravasation, we developed a stochastic model inspired by the persistent random walk. In our model, we have some assumptions. Cells are individual active agents with certain volume. And these cells can move freely in a 2D section with a given speed and persistent, the table that I just showed you. And also these cells get nutrients, oxygen from blood vessels, stochastically distributed. And also cells within those regions around the blood vessel, the red zones, prefer to stay there rather than leaving these red zones. Cells can divide, and the probability of dividing is related to 3 factors. Are these cells near a blood vessel or not? Are these cells surrounded by other cells, or they are like kind of free? And also the time since the mother cell divided, because as you know, cells need time to prepare for the division. So we take into account these 3 factors to let cells divide. And cells that spend the G1 phase in the area where oxygen and nutrients is high, they divide faster. And of course, cells can die if they don't divide during particular cell cycle periods. And the most important part for us is the intravasation, that these cells, during their movement, they can go and intravasate to the blood vessels. So our model is abstraction of a real-life scenario of breast cancer tumor, starting from a single cell. And these cells can divide, migrate, invade, die, and also intravasate. And behind each of these processes, there are a lot of mathematical equations. I'm not going into details for that. And to avoid losing cells going out of the simulation field, we used a technique called periodic boundary conditions. So if the cell will leave from the right side, it will appear again on the left side. I will be showing now some examples of the simulations. As you can see, the upper panel shows cells with low persistence, and the lower panel shows cells with high persistence. Columns refer to different speeds. So the upper left, low speed, low persistent, and right bottom, high speed, high persistent. All the simulations start with a single cell, which will be randomly located. And these cells can move freely and divide. And as I mentioned before, the red areas refer to the zones around the blood vessels where the concentration of nutrients and oxygen is high. And when the cells are there, they get excited, they divide faster, and they prefer to stay there than going out. We run the simulation for 90 days, and we look at the number of cells generated in the end, number of cells that intravasated, the dead cells, and how many cells went to the red zones, and what is the total time. Maybe I'm having now some problems with the presentation because I don't know if you see it clearly, but it sounds it's breaking.
Yes.
Yeah.
Yeah, we see things happening.
Yeah,
It's
maybe
very
because
good.
it's a little bit heavy. So yeah, as you can see from the upper left plots, we see kind of a small cluster, dense cluster, and that because of the low persistent and low speed. And also we see at the center some white areas, refers to necrosis, and the cells are getting more distributed with the increasing speed and persistent until we reach here. So you see the whole simulation field is full of cells. After 90 days of simulation, we looked at the results. Here, different colors refer to different speeds, and the bars within one color refers to different persistent values. As you can see, there is a gradual increase in the number of cells, which refer to or reflect the tumor growth, which means that both speed and persistence are positively correlated with the tumor growth. Maybe you are wondering about why here there is a kind of plateau in the tumor growth, and this is because of the limited area of the simulation field. We tried the bigger simulation fields and we keep seeing transitional increase. But of course, at some points, we will see that plateau. So making bigger simulation fields will make clear correlation. When we looked at the number of cells that intravisate during these 90 days, as you can see, between different colors, there is a gradual increase. So speed is positively correlated with intravisation. But if we look at particular color, let's say the blue one here, we don't see a trend because here we have very low persistent and the persistent is increasing, but there is no increase in the number of intravisating cells, which means that only speed is affecting the intravisation. So from this slide, we can see that fast cells will make a bigger tumor with more intravisation. So changes in cell migration will affect tumor growth and intravisation. However, cell migration is controlled by an organelle in the body called Golgi. And Golgi in healthy cells in normal life, in addition to being a sorting station for secretory pathways and post-translational protein modifications, it controls cell migration. But that when it's healthy and intact, as you see it here in the photo. But in many cases, the Golgi is not intact. As you can see here, it's kind of spread and making several vesicles. So thus, this kind of fragmentation affects its function in regulating cell migration, and how the migratory characteristics of speed and persistence change when the Golgi is fragmented. And this brings me to my main goal of my PhD, which is understanding the pathological relevance of Golgi fragmentation in breast cancer. Maybe you will ask why we think there is a pathological relevance. The answer for this, because in patient samples, we see severe fragmentation, as you can see here, whereas the Golgi is more intact in adjacent normal breast cells. To do more quantification methods, we generated PSDs, PDX. So cancer cells were grown in mice, but they are originally taken from patients. And we stained the Golgi, and we see that high percentage of the cells have fragmented Golgi. As you can see, like about 50% of the cells have very severe Golgi fragmentation. If you look here at this plot, if we add those two, we would not get 100 because, and the reason for this, we ignore the cells that were not very obvious, because sometimes you see a fragmentation, but it's not a real fragmentation. We were very picky and strict when we defined Golgi fragmentation. So these vesicles should be more than 4, and they should be totally separated using the Z-stack microscopy. So more than 50% of the cells have this severe fragmentation. Is that by random? Has nothing to do with the Golgi, with the cancer progression, or it has a meaning because we have seen it a lot. For that reason, we wanted to know why breast cancer cells, most of the cells have fragmented Golgi. To address this question, we studied a triple negative breast cancer cell line called BT549, where the majority of the cells have intact Golgi, as you can see here. So we induced Golgi fragmentation by silencing a gene responsible for making the structure of Golgi called giantine, or GOLG-B1. So we induced the Golgi fragmentation, and we found also that Golgi fragmentation or giantine low expression is associated with low survival. So low GOLG-B1 giantine lead to Golgi fragmentation, and lead to low survival rate. We try to understand why this is happening. So after we did the knockdown, we couldn't succeed in having all the cells having fragmented Golgi. The efficiency of the knockdown was very high, but only 65% of the cells got fragmented Golgi. And the reason for that, because there are several proteins responsible for making the structure of Golgi. Giantine or GOLB1 is the main one, but still some cells can recompensate with other Golgi proteins. We were happy with this, and we used that to see what will happen to the cells when they migrate, when they have fragmented Golgi. And we saw that about 25% of the cells get reduced with their speed. So Golgi fragmentation causes cells to be slower. At the same time, we looked at the invasion capability of the cells with Golgi fragmentation. We did something called fluorescent digestion assay. So we seeded the cells with and without Golgi fragmentation on fluorescent gelatin, and we kept them overnight. And surprisingly, we saw huge digestion of the green area, the gelatin, when cells have fragmented Golgi, which means that Golgi fragmentation increased the capability of the cell to invade and intravasate. And as you can see, there is about 33-fold change increase when the Golgi is fragmented. We took these data and we implemented them again in the model to see what will happen if the tumor has more cells with fragmented Golgi. And we noticed that also after 90 days of simulation, that the tumors are smaller, more condensed, and they make more intravasating cells. And to look at this from single cell point of view, we developed a pipeline to look at the transcriptomic data and examine particular genes and see their level, and also do a lot of extra stuff for differential expression analysis, decision trees, and a lot of work. But now I will be only focusing on one feature of our pipeline, which is called DiskBio, which is the expression of the cells. Here we see TNSE map for 1,500 breast cancer circulating cells. To the left, we see the expression of giantine. As you can see, it's low expressed in about 80% of the cells. Each dot represents a cell. And if we compare the expression of giantine to the expression of a housekeeping gene, actin, you see it's very high. So we confirmed that circulating cancer cells have low expression of giantine. But what is the expression of giantine in normal breast and also in primary tumors? We studied that and we found that giantine is the least in the CTCs, increases a bit in primary breast tumors, and in normal breast tissues is much higher, which means that giantine has a role. And we didn't find that only in breast cancer, but in several cancers. And this indicated us to nominate giantine as a potential biomarker for breast cancer progression. But of course, a lot of extra work is needed to validate those results and to look at different data sets and maybe different cancers. So maybe giantine can be one day a therapeutic candidate that can help in cancer treatment. Let me now talk a little bit about cancer treatment in general. First signs of tumor treatment appeared in the surviving pages of an ancient Egyptian papyrus describing 8 cases of breast cancer treated by burning the area. And that paper says, as for cure, there is none. And since that time until now, we are struggling to find a cure for cancer. We succeeded at some points in some cancers, but still we are very far away from finding a complete cure for cancer. One measurement could be looked to evaluate the treatment, and it is the survival. So survival is more than 3 times higher when cancer is diagnosed early. However, there isn't enough stuff to do the diagnosis of cancer early. So this calls for new solutions or finding better ways to help diagnosing the cancer early. 100 years ago, or even 50 years ago, cancer was often not detected until it was too late. And today, advances in testing, early diagnosis, treatment, and research are helping increasing the rate of survival. As you can see, different cancers have different survival rates. Sometimes it's so high, and as in testicle and thyroid cancers, but also it's still very low in pancreatic cancer, which needs extra work to be done to improve the survival. Let's now talk what are the possible ways of treating and how that is connected with mathematical and computational approaches. Surgery is the oldest type of cancer treatment, and it's still today effective for many cancer types. It aims to remove the tumor and nearby tissue during an operation, and the surgeon often makes large incisions to remove the tumor. But recently, minimal invasive surgeries are applied using robotic techniques, and those robotic techniques are mainly dependent on computational approaches. I will go to that later on when I will be talking about artificial intelligence. The second main treatment is radiotherapy. It aims to destroy cancer cells and leaving normal, healthy cells unaffected. Radiotherapy requires careful planning and targeting the area to be treated accurately and to minimize killing healthy cells. This accuracy also means that higher doses can be given safely. The third cancer treatment, which is the most widely used one, is chemotherapy, where a heavy drug is used, and this drug will be given maybe as tablets or in the veins, and the goal is to stop cancer cells reproducing, which prevent them from growing and spreading in the body. And of course, the effectiveness of the chemotherapy varies a lot based on the patient and the cancer type and on the drug itself. And also here, the power of mathematical modeling can help in improving the chemotherapy. Immune therapy is one of the most important revolutions in the history of cancer treatment. Our bodies are equipped with cells that form an army to defend itself. B and T cells are programmed to kill invaders, including cancer cells. One of the immune therapy methods is to create vaccine using neoantigens, which are antigens that can be selected by identifying mutated proteins in the tumor. The vaccine can be activated in the patient, activating the patient T cells, and then the T cells will go in the body, find their target, and kill the cancer cells. Another approach is to train the T cells We take the T cells from the body, educate them, train them in the lab, and then send them back to the body so they can, so the patient can have a stronger immune and in some ways better fighting to the cancer cells. Immunotherapy drugs have been approved to treat many types of cancer. However, immunotherapy is not yet as widely used as surgery, chemotherapy, and radiation therapy. Hormone therapy is also used, and it's only can work when the hormones are the main player in the proliferation of the cells. Cancers that can be hormone sensitive include breast cancer, prostate, and womb, and ovarian cancer. So it does not work for all types of cancer. Targeted therapy uses specifically designed drugs such as monoclonal antibodies that act on the surface or inside the cells. Tests can tell us if those cells have those particular targets for the drugs. For example, Herceptin is an antibody drug that targets and slows the growth of HER2-positive breast tumors. These tumors have high amount of HER2 receptors, which are the target molecules of Herceptin. The last type of treatment is called the stem cell transplant, which can be used to treat certain types of cancers, such as leukemia, myeloma, and lymphoma. The bone marrow is a soft sponge tissue. It contains the blood-forming cells. In leukemia, for example, the patient bone marrow makes abnormal white blood cells, and over time, these abnormal cells crowd up the production of healthy white blood cells. In stem cell therapy for leukemia, healthy blood cells from the patient itself or a donor will be collected, and then these cells will be frozen to be used later on. Then the patient will receive very high dose of chemotherapy to kill the abnormal cells, but also the therapy will kill the normal cells in the bone marrow. Once the abnormal cells are gone, the healthy blood forming cells are transplanted into the patient again, and then new white blood cells, normal white blood cells, will be made. For some cancers, the best way is to combine several types of treatment, such as surgery, radiation, and chemotherapy. Combined treatment has shown very good results in pediatric brain tumors, where the survival of the patient who had combination therapy of chemo and radiotherapy is significantly higher than the one who got only radiotherapy. Not all cancer patients who will receive the therapy will respond to the treatment. Respond or not respond, that is indeed the therapeutic question. But why do some people respond to cancer therapy and some don't? One explanation is that cancer is heterogeneous genetic disease, and the response to the therapy is not as systematic or clear-cut as simple matching mutation to a treatment. Other factors can also play a role in the response, and recently they found that the community of microorganisms living in the gut can play an important role in the response. Cancer cells can become resistant to treatment, so the cancer comes back after the treatment, and doctors call this recurrence. Recurrent cancers can be in the same place or same place where the cancer first originated, or they could be in another part of the body. Cancer recurrence by itself can mean that cancer is much more complex than we have been treating it. Can we ever cure cancer completely? According to the Time magazine, there is no way to expect a single hero who can cure cancer, but many heroes. A cure for cancer might be on the way, but that way is so long and complicated. It involves several interacting, looping stages, starting with basic science research and ending with a lot of clinical trials. Much of cancer research is done in biological systems. Takes time and it needs a lot of money. This line of research is very essential and great, but it has a lot of limitations, not least time and money. To make the way to cure faster, we need a team that consists of several players. Pathologists, oncologists, pharmacologists, biologists, and also bioinformaticians and modulars. And to reach effective cancer treatment, we need all the players on board. But how math can help curing cancer? The complexity of cancer is well suited to quantitative approaches, and it provides challenges and opportunities for new developments. The advances in the technology have resulted in a huge amount of biomedical data. Big data is not just number of individuals that are in the samples, but is what we can actually collect. So from one drop of blood, hundreds of gigabytes of omics data can be collected at different levels. Some of these data will come from proteomics, the study of proteins presented in the sample, and the proteome is the counterpart of the genome. Together, the genomics, proteomics, metabolomics, and transcriptomics are producing a tsunami of data that can help finding better therapeutics. In addition to omics, there is much clinical data, such as the MRI scans. This huge amount of data is too large for a single team or even an army of scientists and clinicians to analyze and find patterns without the help of math and informatics. The secret for reaching a cure is by linking multiple layers of clinical and molecular data together. Here comes the job of mathematicians and bioinformaticians to apply complex math and create algorithms. Such data integration may lead to a precise diagnosis and help identifying specific gene networks involved in a target in a drug response, leading to better therapeutics and therapies. Math has long played an important role in cancer research and treatment, but not only for measuring purposes such as the diameter of cancer lesions in melanoma, for instance, but also to estimating the survival of the patients and under different conditions of the treatment. And also math can help predicting the treatment response and evaluating the results statistically. Math has the power to lead us to understand how cancers grow and how cancer cells function. This knowledge equips scientists with new tools to predict and in turn improve cancer treatment overall. Accurate predictions of tumor growth are key to determining the right dose, as I mentioned before, for radiation and chemotherapy, and how often the patient should undergo screening, and also whether a treatment is effective or not. Considering the cost, human effort, time, ethical issues involved in the clinical drug trials and basic medical research, artificial intelligence and mathematical modeling can potentially contribute to the discovery of better cancer treatment regimes. When it comes to artificial intelligence, it's much more than just robots. It's a wide-ranging tool that enables people to rethink how we integrate information, analyze data, and use the resulting insights to improve decision making. One way to do this is through machine learning and deep learning. I will give example now. The whole mammography machine represent the artificial intelligence. The computer on the left represent the machine learning, whereas the algorithm that will analyze the mammograms is the deep learning. So the deep learning is a subset of machine learning where neural networks are developed to allow the machine to learn and resolve the problems as a human brain. One example of neural networks is the artificial neural network, which consists of three layers. Input layer, where we get the information in different ways. The second layer is the hidden layer, where the mathematical and the computational work is working or happening. And of course, the output layer, where we get out the results of the artificial intelligence. Artificial intelligence is believed to have a dominant impact. Here we will see how they moved from how the brain is working and to mimic it in computers in silico to do something similar to the brain but at higher levels. So what we cannot do in our regular brains, we can do that with the artificial intelligence. As I have mentioned before, it has a lot of applications in clinical situations and also in the trial to fight cancers in different levels, starting from the management to the diagnostic with the pathology, and also drug discovery, and also recently with the precision oncology. Let's see an example of artificial intelligence. As I mentioned before, with the robotic surgeries, artificial intelligence is highly involved because the artificial intelligence will translate the movement of the surgeon fingers and hands into precise movement inside the incisions in the patient. And the math has very important role in controlling this process. Let us see now how the deep learning works. For example, artificial neural network is used to help staging lung cancer via X-rays. For that purpose, we need two data sets, one data set for training and one data set for testing. First, the network needs to be trained. X-rays of cancer patients are fed to the system. Initially, it struggles to correctly identify the condition, but every time an X-ray is fed in, the network structure is adapted and the diagnosis ability improves. The more the training, the faster the identification becomes. It takes thousands and thousands of clinical data sets to train the machine. Only after the network is optimized, it can correctly identify an unknown X-ray from the test dataset. Computer-aided detection in mammography is routinely used by radiologists to assist with the image interpretation. These programs typically use handcraft features to mark sites on the mammogram that appear distinct from the normal tissue. And for that purpose, they use more developed networks than the artificial neural networks. They call it convolutional neural networks. At the moment, there are a lot of types of these convolutional neural networks, which differ mainly with the amount of layers they have and the needed time for the training. But how accurate is the artificial intelligence compared to oncologist expertise? Reading X-rays correctly is a complicated process, but artificial intelligence is making it fast process, and especially when it comes to simple images. So computers have passed a human capability a lot. But when it comes to complicated 3D images, both human and artificial intelligence showed almost same efficiency, but from different angles. Let me give you an example. At New York University, a research team analyzed images collected as part of a routine clinical care over 7 years, and they created a very huge data set, and they used it to train the machine learning. And they developed a special convolutional neural network. And by the way, that network or that data set contains more than 1 million images. How it works, so their artificial intelligence identify related patterns in the data that radiologists couldn't see because the artificial intelligence go part by part in the pixels to see if the tumor or the sample is more benign or cancerous, generating 3 heat maps. What they found that the computer detected cases radiologists couldn't do it, and the opposite at the same time, like the radiologists detected stuff the machine learning couldn't. So they clearly said that the goal here is not to replace a human radiologist, but to combine. So we get a higher effective diagnosis and more precise. According to the global market Insight magazine, the healthcare artificial intelligence is dramatically increasing, and currently drug discovery is holding 35% of the overall market share, which means that the drug discovery is highly important. The huge amount of experimental data accumulated from the past decades is a valuable source for the success and failure of compounds in the entire discovery process. Machine learning can increase the success rate of predicting future drug candidates in the discovery, experiments, and clinical trials. There are 2 types of machine learning methods, the unsupervised and the supervised. The unsupervised is used for classifying the terms that we are looking for, let's say the drug components, or the supervised one to classify if they are targeting well or not. And those kind of machine learning are widely applied at the moment to help the companies to find target drugs when it comes to using single cell omics by looking at biomarker discoveries and finding a potential target for particular case. Mathematical modeling is a process that uses math to represent, analyze, and make prediction. So from that case, Pfizer is using a lot now machine learning and math. They are collaborating with a lot of informatic companies. Some of these collaborations didn't work properly, maybe because it was more oriented in informatic way without getting more biological insights. And now they are working more with the people who are expert in bioinformatician rather than just informatician, and trying to find candidate tools for new therapies. As I mentioned before, there are several ways to include math in the process of diagnosis and treatment. Artificial intelligence is one of them, and the other one is the mathematical models. But how the mathematical modeling is working? As I mentioned before in my model, we need to depend on math because math has one interpretation. But when we use words, many people can understand the same sentence in different ways. So the goal of mathematical modeling is to change the sentence, the logic of the sentence, into very precise mathematical formulation. And we do that by changing the assumptions, if you remember the 8 assumptions that I showed in my model, into formulas. So those formulas will be used, and before that, we need to know what are the variables in our model. So we have independent variables and dependent variables, and of course some parameters. The parameters have fixed values. We call them constant values. The goal of moving from imprecise knowledge to precise knowledge is to get better understanding of the system that we are studying. And of course, we need to find solutions. And those solutions can be done by analyzing the outcome of the models. And that could be also return us back to the mathematical formulations if we found that some of our outcome does not match with the reality, which means that either we have a gap of the information or our assumptions and the translation is not correct. So running the analysis several times and repeating the process can give us a good way to look at the models, but of course we need to validate the models using experiments. Mathematical modeling is not what things are. When we look at mathematical modeling, we look at relationships between the elements that we are studying. So from what is common between those two pictures? Maybe from biological point of view, they are totally far away. We see here lion and prey, and on the other one we are seeing immune cells trying to attack cancer cells. From mathematical point of view, both are the same because those two systems are having predator-prey interaction. So the prey here could be the cancer cell or the mammal that would be eaten by the lion. So understanding such interactions can help us to understand the secrets of cancer cells. And this has been studied a lot, the prey-predator system, and it was applied a lot in immune systems and also in the immune therapy now. Some new papers did not like this idea, and they said, no, it's more complicated than simplifying it into this way. And maybe that is the reason why we are not able to reach very high response. So better mathematical models than the prey-predator system should be applied. As I mentioned before, that mathematical models can help at the moment in the clinical research and can help us to get better decisions. For instance, tumor growth or estimating tumor growth can be affecting a lot the decision for taking the treatment and which treatment we should apply. The biology behind tumor growth is really, really very complicated and deep. But when we look at the outcome of most of the mathematical models of tumor growth, we see something similar to this line. So the complexity of biology behind tumor growth is not shown in the simple outcome of the mathematical modeling. Most of the mathematical models are dependent on the classical models developed several hundred years ago. And one of them is the mathematical model, GAMPI. And as you can see here, it's recently used to evaluate a treatment in mice. And they used this simple mathematical model. But how did Gompertz come up with this model? In fact, he was not interested at all in cancer. He was interested in human death. He wanted to simplify the ways of calculating how many people are still alive in a certain age range. So he looked at life tables here, and he found two patterns. First pattern, for certain age ranges, the death rate did not change with age. So he converted this into this equation, and by solving this equation, we will get a number of people just declines exponentially. The second model, which is the one that is now used as the most common model for tumor growth. So in this pattern, he saw in some cases that the death rate increases with age. So he wrote down a form of increased death with age. And he found that the number of living people starts off here. I don't know if you see the mouse. Starts off and increases faster than exponential. So this is the concept that ended up transforming the way people modeled tumor growth. And how do we get from this to this? It's very simple. It's just like flip-flop, because here we're talking about death, and in the tumor growth we are talking about the number of cells generated. Gompertz model for tumor growth is heavily used in literature, and it inspired a lot of new models. For example, in this paper, they developed, or they found, based on mathematical modeling, that the algorithmic treatment in chemotherapy and radiotherapy is more effective. And they proposed a new radiotherapy treatment using logarithmic late intensification. The beauty of and the power of mathematical modeling lies in its potential to bridge the gap between in vitro and in vivo experimental strategies. And in order to achieve more effective treatment, let's say in pancreatic cancer, for example, they used mathematical modeling to bridge the gap. So what they did here, they used this drug is showing a huge effect in lab in killing pancreatic cancer cells. But when this drug is used for patients, the survival rate is still very low, which means that the drug is not working. So they wanted to understand why the same drug is working in lab, but in reality or in vivo experiments did not work. So they ran the model and they used estimated the parameters from in vitro experiments. And when they applied the same settings in the mathematical modeling, but instead for 2D, in 3D, they found that the drug is not able to kill the cells, and the cells become resistant. So in that paper, they predicted that in vitro is not matching with the in vivo. And when they did the experiment in vivo, they found that using the same concentration of the drug did not kill the pancreatic cells as it did in vivo experiments. And that can give an explanation why a patient with the pancreatic cancer are not getting well treated using this drug. Back to the immunotherapy, which is now heavily used in research, and maybe new insights showing good results in clinical. We see that it's heavily published now in the literature, and the amount of also therapeutic drugs got approved is so high with immunotherapy, but the response is still very low. And this needs some more work to understand what's happening, why many patients are not responding to the immunotherapy. Let me end with a work by the Moffitt Cancer Center, where doctors, researchers, and modulars are working together to develop a new way of treating cancer using the personalized cancer therapy. And they called it the Evolutionary Therapy. In simple terms, they are depending on delivering the right therapy at the right moment. Using a very basic idea in science by Darwin, because Darwin provided the recipe for evolution that requires just variation and selection. And in cancer, these two elements are there. So the thing that they do at Moffitt, that they combine evolutionary principles with mathematical modeling to predict how these cancers can evolve and grow, and then we can potentially manipulate their evolution and kill them. I will show you now an example of a mathematical model done at Moffitt, where we have two now treatments. The left one, it's a continuous, so the treatment is given continuously, whereas the right one is off and on, so they don't give it continuously. As you can see in the beginning, the continuous treatment did not shrink the tumor as the off-on, but then what happened that the tumor, sorry, I missed, the left one is off-on and the right one is the continuous. Let me play the video again. So, yeah, as you can see here, with the continuous treatment, the beginning, the tumor shrank a lot, but then resistant cells appeared. Whereas with the on-off treatment, they can control the growth of the cells. So far, they couldn't control it 100%, but they keep it limited. The good thing about this modeling is not only in silico, but they also are trying it now on some patients. And this is giving very promising results, and that could be something applying in the clinic very soon. Let me end with giving some take-home messages. The complexity of cancer is well suited to quantitative approaches. Artificial intelligence and mathematical modeling contribute to cancer treatment and helping to elucidate mechanisms, and by providing quantitative predictions, and also validated experiments are needed. In the end, let me say that mathematical models can complement experimental and clinical studies, but also challenge current paradigms and redefine our understanding of mechanisms driving tumorigenesis and the treatment of cancer at the moment. Thank you very much, and if you have any questions, please go for it.
Thank you very much, Salim. It was a really fabulous presentation, and all these animations, it was fantastic. And it's very understandable, if I can say. But I'm afraid that we are out of time, and it's impossible to do. Even for myself, I wrote several questions and comments, but I cannot let us go this way, unfortunately. And I would like to...
Let me step in.
Yes,
Let me step in. Can I have a suggestion? So, I can start speaking at half past 15:30, and then I will speak as long as you let me. How long time do I have? What is the end of the seminar?
No, because we're completely out of time. Let me see what do we have. We should finish at 5.
Yes, yes. So see,
now it's
this
4:20 already.
This is no problem at all. And in fact, Salim got a number of things that I wanted to present in my presentation. So I just take them off. So I suggest now that we take 10 minutes. Those who have questions, please discuss with Salim. The others may take a little break. I will start in 15:30, and I will speak 30 minutes. Okay?
Okay.
So I'm very curious to hear your questions.
Yes, even 40 minutes we can give you. Yes.
Yes, we are fine. We are fine.
So maybe in this case, if somebody has questions to Salim from the audience. No.
There was a comment. Thank you for your presentation. I would just like to add that the FDA has approved an AI software for prostate cancer in September. It is used to help the pathologists diagnose malignant samples. However, it was not very beneficial for the benign ones. Hopefully, there will be more programs soon. All the best, says Maria Levkova. Thank you, Maria.
Thank you, Maria.
I have several questions, but maybe I will start from your first part with your data. When we discuss the speed of the cells and persistence. So I understand from your model, you found connection with cell density and vessels, right? Is it correct?
Yes.
And if in your model, first of all, about which cells we talk about the malignant cells. Yes, it's these slides. We talk about malignant cells, or if it will be I don't know, any feeder cell culture of other type of cells, the cell density is also plays a role, or it's only presence of transplanted cells.
Yeah, let me clarify one thing. When we built our model, this model, we built it based on our experimental data, which were basically or mainly on triple-negative breast cancer cells. But later on, my supervisor said we are providing very narrow case, and for that reason, we need to make it wider and make the model more versatile, so people can use their own data by changing the way of the cells divide, how the cells migrate, the density, the time spent or the doubling time and everything. So now on our GitHub, we have the models which are very versatile. So people can just change the numbers based on their experimental data. And they can run the simulation. They don't need to run it for 90 days because that needs supercomputers, but they can run it for like shorter time because, as you know, it will go exponentially. The more the time, the more the cells, and the more the computational power. So it's not limited for particular settings. What I showed, it's limited because we are interested in breast cancer and Golgi fragmentation. But what we have on GitHub, it's versatile for any type of cancer, different types of movement, and also for cell division and density.
But you was responsible for mathematical description and mathematical model and all this very nice, I don't know, results is just mathematics. And do you know if, as I understand again, but do you know the experiment itself, what they took breast cancer cells and put them where? To the flask or to the animal, or it was, I don't know, some, how they mimic metastasis in liver, or that's why I ask about cells density, because another question is which kind of cells and how metastasis behavior in real tissue.
So I was not only responsible for the mathematical part, but also the experimental. And that, I think, the thing that I like in my PhD and also now in my postdoc, because there is a combination of experimental work and computational work. Yes, we did most of the experiments in vitro in 2D flasks, but we estimated the parameters, or we scaled the parameters in 3D, because in real tumors, cells are not moving or dividing in 2D, but in 3D. We know that is limitation of our model, because we started with 2D experiments, but we scaled the parameters to be in 3D. But we had to do this because using 3D experiments is a little bit complicated when it comes to for breast cancer, because we tried that in the beginning, but we found that there are many factors affecting the stiffness, of the scaffold, and what kind of scaffold we're gonna use. It affected a lot the results. So we decided to start simple in 2D. And what I know that now other PhD student is taking over, and now he is trying to do the experiments in 3D. The main goal was to build the model so it can take the parameters. We started with parameters from 2D, but at the moment they are now doing it in 3D, and hopefully very soon we will place those parameters or the values in the model and see if that will give us some results. And I think that will be very interesting for us to see the differences in 2D and 3D.
Yes, very interesting. Then we have another question in comment in chat from Dr. Maria Levko. Maria, maybe if you are here, you can pronounce it and ask Salim to respond. Hello? Or I can read it.
I did read it.
Ah, you did it. Okay,
Yes.
okay. Yes, I also read it now. So, Salim, do you see it? So, thank you for your
No, I don't see
presentation.
the question. Yeah.
There is, I don't know, somewhere you can open chat. And the last, thank you for your presentation. I would just like to add that the FDA has approved an AI software for prostate cancer in September. It is used to help the pathologists diagnose malignant samples. However, probably it was not very beneficial for the benign ones. Hopefully, there will be more programs soon. All the best. So if you know something or you have comments about this, I don't know. Unfortunately, there is no voice of Maria. We should start in 1 minute. Let me also leave my okay. Maybe.
So if you, Salim, stop sharing.
Yeah.
And then I really like to thank you once more for not only stepping in, but presenting such a fantastic talk. Thanks a lot. And to Thomas as well, who I think
Yes.
here becomes this time of that people need to run to kindergartens and things like that. So maybe he turned off, but thanks to him too. And then, I don't know, those who need to stretch their legs, there's 1 little minute left.
The problem, I don't see where to stop sharing. Okay, yeah. I found it.
Yeah.
I was looking the wrong place.
Let me see now if I can find where to start sharing.
Is really fantastic. Yes, I would say.
Thank you very much. And sorry, I took extra time, but honestly, I didn't practice, so I didn't know exactly how many slides I need to put or take
from the very
off.
beginning today. From the very beginning today, we're out of time, so it's.
You did very well, Salim. Thank you. I know.
Not too many people here, so it's like seminar. It's not a big conference, so in this more intimate contact and situation, we can be flexible. I think it's okay.
That's good.
For me, I just afraid that Christina will be limited in 30-40 minutes. And Christina, what happened? Can you explain?
You can just. I'm fine.
I know some people who call me in different name, but what is interesting that they in most cases when other people make mistake in my first name, they call me Yuri. I don't know. There is no one, maybe R. No one single letter similar to Yuri, but in different heads there are some, I don't know, some irrational connection. So okay.
This is totally fine. So I'm having my watch in front of me.
Yes, let's go ahead.
I will give you in some way a sum up of everything we spoke about. Some things maybe I will go through a little bit faster. And it will be about the genomics of breast cancer. So we are all, as you saw, so Salim joined us very recently from a computational group or from a group of biostatistics. But me and Xavier, who I hope will be able to speak to you next time, and Thomas, and also hopefully David Kunke, another senior scientist in my group, who is an expert on polypotent stem cells. Hopefully with you, Igor, he can organize another course. And a guy called Stefan Boss-Haugen, who also is a senior scientist working with genomic variation. We all, in one way or another, focus on breast cancer. And so everything that I'm going to say, just like Thomas did, will be focused on breast cancer. But of course, all the methods and all the observations and all the analysis is very relevant for other types of cancers as well. So just a few, just so that you can see the things visually, like Salim was so good to present things so that we can imagine them visually. The breast cancer is a disease which happens in the breast cells. In 80% of the cases, it happens in those ducts, the so-called tubuli, where actually the milk, when it is produced in these cellular structures here called the lobes, is running through down to the nipple and out of the breast. So when there is an abnormal cell growth, and it grows and grows and grows inside the duct, we call this ductal carcinoma in situ. So the carcinoma is still inside the duct. That's why it's in its place in situ. And why exactly do these cells grow inside and not outside? And what exactly keeps them inside until it keeps them inside is a great area of research. A lot is being done on that, especially the other way around. What actually does make them break through? Because when they break through, they go through this basal membrane and they turn into invasive cancer cells. So they can spread locally in the breast around, but also through the circulation into through the lymph ducts or through the blood vessels. They can invade both the bone marrows, the way I described yesterday with our single cell bone marrow study, but also distant organs to form metastases. And now we have been studying these cells both in a bulk, as shown here, but also in a single cell, like Xavier and Salim are going, hopefully until we meet next time, if we meet next time, generate data to show you what is the state. They already have quite a lot of data. What is the state of all these cells separately? So if we look at one cell, we at present collect data on several different levels. So here is the nucleus. In the nucleus is the DNA. There we collect data on 25,000 genes. Actually, we do now, if we work with arrays, that will create 1.6 million probes. If we work with full genome sequences, as we've done in the last couple of clinical trials, we will actually have a lot more data points. Then, of course, these genes are transcribed into mRNAs and microRNAs, which leave the nucleus and out into the cytoplasm. And we do screen for them. We have an array of 1,800 microRNAs for which we can characterize these tumors. And we have arrays for 60,000 mRNAs, but more recently we also do actually whole genome transcriptomics. So we sequence everything which is transcribed, which creates at minimum other 60,000 data points. So the proteins are, of course, translating to proteins, just I showed into my introductory lecture yesterday. And those we can study in different ways, not as high throughput as transcriptomics, but mass spectrometry methods and array-based methods allow us to at least study a couple of hundred proteins at a time. At the same time, we know that in the genes, there are these switches of on and off, which Thomas described in very big detail before me, which condition whether a gene will be expressed or not, or whether there will be coming an mRNA out of it into the cytoplasm or not. And I'm using this to show that for one single tumor, for one single patient, we generate data points as based on our previous generation analysis, which was array-based analysis. 1.6 million data points at the DNA level, 450,000 data points at methylation level, 60,000 data points at mRNA level, 1,800 points in microRNA levels, and something like 200-300 proteins. And this is for one patient. And we have these data for 2,000 patients now. So the idea or the question is, how can a simple mind and how can a biologist take in all this data and make something meaningful out of it to identify, when we decompose everything into single genes and single molecules, and we start to connect these single molecules into given networks, how can we again reconstruct the cellular networks and what is normal in the cellular networks and what is normal and what is abnormal and what causes a disease into one or another organ. And I'm very grateful to Salim, who actually departed from the single cell topic to describe many of the mathematical models which are used to in fact indeed integrate this information back into some biological meaningful system. We summarized some time ago these principles of integrated genomics by showing how we in Oslo have organized our breast cancer research. It has been our ambition for every woman who presents with breast cancer in the Oslo area. So this is both Norwegian Radium Hospital, Ullevål Hospital, and Akershus University Hospital, the three largest hospitals in Norway. For every patient, to be able to collect blood samples. Blood samples we have been using for genome variation studies, which I described yesterday, but also take a sample of the tumor, which we can then subject to both creating images, a very classical histopathology, but also isolate DNA, RNA, and protein so that we can do the experiments that I showed in my previous slide, so that we can study all the genes in the genome of that tumor, their expression in transcriptomics, and what proteins do they give rise with. And then with a very nice group in Trondheim who are very good into metabolomics, we have also added that to see which tumors do have glycolysis, in which tumors do we have active metabolism, and all that. And this indeed generates this huge amount of data, which now our big challenge is to find out which of these data can create parameters that could be eventually useful in a clinical setting. So can we say, out of all these measurements, if we put them together, what could be important to know for the clinical outcome, or for a given response signature to a drug, to a given adverse side effect signature, which becomes more and more important the longer patients survive and the longer the adverse side effects can function. And all this, in a way, if we create this sort of a barcode or some digital output for every patient, which can actually guide therapy, would benefit personalized treatment. So what is the situation in breast cancer? We touched upon it on several occasions. So breast cancer is a little bit simpler than other diseases in some respects, in other cancers than some respects, because there are these two receptors, actually three, also the progesterone receptor, which may or may not be on the surface of the cell. So this is very useful both diagnostically and therapeutically. If you have on the tumor cells the expression of the estrogen receptor, you could actually target it very successfully with hormonal treatment, just as I showed yesterday in this clinical trial that I presented. If you, however, do not have that, then it is a little bit difficult, because you cannot target something that is not there, and you cannot target something that you don't know what it is. So that is exactly why the so-called triple negative cancers, so these are the ones that do not have the estrogen receptor, do not have the HER2, and do not have the progesterone receptor, which is missing here but should have been here. They are called triple negative, and they do not have very good therapeutic offer. So these are the ones we are mostly working with now. But those, as also Thomas showed, or Salim as well, those that have those expression, these receptors on them, they actually respond quite well to treatment. And this is in a way a success story for breast cancer. It makes it easier for us to study it, because here, very rarely in the case of medicine at all, we know what is the reason for proliferation, we know what is the reason for cancer, and even when we target it, we get exactly the expected effects. Indeed, we can stop it through that. So this is the perfect targeted treatment, which has been known since the 60s, 70s, and which is so desired in other cancer forms, but so difficult to reach where we don't have clear targets. Also, we know that these different subclasses of breast cancer have different survival. So early in the 2000s, we could recapitulate these results and even look further by the first whole genome expression array. So the first whole genome analysis, meaning the analysis of all genes together, was at a transcriptome level, and this was just historically because the transcriptome was the first that Pat Brown and David Botstein managed to develop from northern blot with 20, 30 points to 10,000 points and cover all the transcripts in a cell. So with this technology in the early 2000s, it became possible to actually study the expression of every gene in the cell. So you could isolate DNA from your sample, isolate RNA from your sample, and then hybridize it with different immunofluorescent probes, and then see which gene is on, which is off. This revolutionized science. Until then, everything was done gene by gene, and now you could look at everything at the same time. So, of course, once you can look at everything at the same time, then you need to think, how do I develop methods to see what I'm seeing? Well, how can I get together the result of 60,000 genes expressed together? Well, one way is, some of you maybe are familiar with this, so-called unsupervised clustering, or the method of the nearer neighbor. If you have 2,000 patients and 60,000 data points for each patient, so how do you compare the 60,000 for patient 1 with the 60,000 for patient 2, the 60,000 for patient 3? Yes, you look at the values in which you can sort all the data in all patients so that you can identify the spots or the genes for which you have the most similar number. And if you do this vertically and horizontally, you can actually sort both your patients, which are now in columns here, and your genes, which are now in row here, according to how similar they are to each other. So all these patients which are in this red block here, they are all very similar to each other along the entire genome, all the genes that you include, or all the genes that you have chosen to include in your study. And you can very easily see what these similarities are. They are because here I have this very big red square in all these patients, and here I have this very green square in all these patients. And here I have another very red square in all these patients. And this tells me, this kind of checkboard pattern tells me that all these cells have very strong epithelial markers. You can see the keratins, the c-KIT, the FOXC1. All they are very low on estrogen receptor. They do not have any estrogen receptor expression, green, very green, and they are all very high on proliferation. So this is a new definition of the so-called triple negative ones. In contrast to them, the hormonally active ones, the one that we knew already from the 60s, have estrogen receptor expression. They have a very strong, you see here, very strong red cluster, which is conditioned by the estrogen receptor, but also by a number of other genes. And actually these ones, GATA3, FOXA1, XBP1, we have seen that they are downstream of the estrogen receptor. So here we have a very clear signaling. We have the estrogen receptor at the surface of the cell, and we have all these other transcription factors that once the estrogen receptor is activated, they start active too. And they create this cascade, which makes all these genes here to be red. So this, maybe you could say, recapitulated the histology results from the previous slide. But we see something more. We see that those estrogen receptor positive ones, which are red here, some of them are more hesitant to be very red. You see that some green appears here. And those, in addition to that, not only being so convincingly red here, they're actually red in the proliferative cluster too. In contrast to others who are just green, they don't proliferate. So the curse and the good and the bad thing about estrogen receptor positive or luminal A cancers, as we call them, is that they do not proliferate. This is difficult experimentally. All cell lines that we want to mimic those, how do you have a cell line that does not proliferate? Also, hormonal treatment works very well on them, but maybe this ability to not proliferate, or this kind of, that they're not dependent on proliferation, makes them so dangerous. They can be dormant for many years and then come with a relapse. But most importantly, this very light blue line here, the luminal Bs, they were new discovery, something we didn't know about, that there is a fraction of these estrogen receptor positive ones that, in addition to hormonal therapy, need also chemotherapy, because chemotherapy works very well on proliferating cells, and they have this proliferating cluster. Another also success story, which was not there when the first array was produced, is the expression of the HER2. So when the HER2 expression was discovered, then very quickly the antibody followed, and the HER2 subtype turned from one of those with the worst prognosis. You can see the pink here is quite bad. It is comparable actually to the basal, like the triple negative that I'm saying, that do not have good therapeutic option. But after the discovery of the HER2 antibody, for many patients, this became a completely game changer. They were suddenly completely rescued from the disease. However, in others, this is still a problem. So resistance is still a problem, even for the drugs that work. And then Xavier, who got sick and could not speak to you today, he also revisited this data in a way and looked at, okay, is there anything more there? And he discovered very recently that in addition to those clusters, which are very strongly dependent on the estrogen receptor, as I said, and the proliferation and several markers of that kind, there are also immune clusters, which you can see in the breast cancers. And these immune clusters can be divided according to whether they have low, medium, or high immune infiltration. And what was interesting in these results, that was in a way that was by that time known, that immune infiltration is associated to prognosis. I mean, if you have a lot of immune cells, that's good because chemotherapy works and prognosis is good. But what he found, which was a bit intriguing and we are still working on, is that if you see here, the cluster which actually has the worst prognosis is not the one with the least infiltration. It is actually the one with the median infiltration, the one that has not the highest and not the lowest amount of immune cells, which brought us to the idea that it is not the question of how many immune cells you have infiltrating, but it is a question of which exactly immune cells are infiltrating. And so he went first computationally to deconvolute data of whole genome expression and to see which immune cells do I have in this tissue. And he could see that, and he would speak to you about that now, that in the different subclasses of breast cancer that I just presented, the luminal A's, luminal B's, basals, HER2, and so, there is a very different composition of immune cells. And this got him to the idea. So for this, he first used also methods that use machine learning. So these are methods that use so-called golden standards. I mean, you look for genes that you know are expressed in the B cells, but never in CTLs and never in Th1 and never Th2. And these are your ideal BCL2 marker cells. And the same for CTLs, the same for Th1s and so. And then you can use machine learning to boost this signature and see, can I get out of this, out of everything, the full compendium of expression data that are out there in the world, can I make these 10 genes into more genes that are markers of the B cell? So you can see that they can grow from 10 to 100. It worked well for the B cells. From 4 to 82, this worked very well for the CTLs. From so and so. So Th1 and Th2 were the ones that were actually very, and of course, the closer immune cells in lineage are, the more difficult it is to get the machine learning to separate them. So that is why he also went further into attacking all this with single cell RNA sequencing. And he would show to you that he has taken tumors and looked for all these cell populations in these t-SNE, so-called t-SNE plots. And this is exactly what the postdoc of Salim is going to be. Together with Xavier, they're going to be looking at these cell populations and see what do they mean for classification, what do they mean for treatment response. And now for first time, we can see all these markers that we used in the classification in the previous slides that I showed you, from which exactly cells are they expressed, in which exactly cells are they produced. So why is this important? Because, for instance, the type of immune cells that you have in the tumor have an extremely important influence on what will happen with this tumor. I mean, if you are lucky, if a patient is lucky to have tumors presenting strong antigen signal, and this tumor antigen can get into the dendritic cells and then get digested by them and expressed on the surface of the dendritic cells, and then can activate your resting T cells, which can get activated into T cells. They can go into the tumor and produce perforin and granzyme to destroy those tumors. And this is a loop which all immunotherapies at different level try to strengthen. Because the problem is that it doesn't work, right? If it was like that, if this was working, there would be no cancer. But the problem is that, first of all, very few tumor antigens do manage to activate the dendritic T cells. And then those dendritic T cells, again, need to have the exact ways to activate these resting T cells. Again, the immune system has this system to make sure that things don't get overreacted. So one can have this alternative scenario in which, so this is when it works and eliminates, but you can have this internal alternative scenario where you actually your neoantigens do not produce that. where in fact the tumor has a lot of inhibitory signals that do inhibit both NK cells and T cells. And that, now Igor is on my way to read my own slide. Then you see that you have regulatory T cells and myeloid-derived suppressor cells that actually do suppress the immune response, which is also a normal a mechanism that hampers autoimmune diseases. But in this case, the tumors, they kind of hijack these systems, and they can use them to not only not get destroyed by the immune cells, but also they can maybe use the immune cells and some macrophages to penetrate into the circulation and actually disseminate. And then tumor grows. So many different processes take part in that, not only expression, but also DNA methylation, which I'll now just not say anything about because Thomas had a whole lecture about, and about how his results contribute to this classification. Here, I just want to show you one funny thing. Thomas did one really funny thing. He did all the mRNA transcripts, 40,000, and all the methylation spots, 450,000, and did a correlation plot. And you may say, this is insane. Nobody does a correlation of 450,000 things across 40,000 other things. But he did this 4 times and got exactly the same thing, that certain CPGs, so you can see that these now what is moving around here, these are the CPGs on all chromosomes that are associated to the expression of different genes on other chromosomes. And you can see that there is a certain subset of CPGs that are always associated to the expression of certain subset of genes. This is not random. And this led them to the studies that he described himself to show that there are a couple of biological pathways. 2, one is the estrogen receptor signaling, and the other is the immune signaling, that are very strictly regulated by methylation. But he spoke about that, and I'm not going to repeat that. But it only illustrates to what degree, in fact, epigenetic studies can pinpoint. They're like a print of a fossil. So you don't see the animal, but you see the skeleton. And the CPGs, they do show which exactly parts of the genome has been turned on and off. And in this case, the estrogen receptor itself. So actually, in his studies, he did explain how the estrogen receptor works, because the estrogen receptor itself is epigenetically regulated, and it does associate to all its downstream genes through epigenetic regulation. We don't say more about that. And this is capable to actually do classify patients. And it is not only in breast cancer, but now they show with his master student Jürgen that it actually does that in all sorts of cancers. And now we can look at these studies that come from the whole genome, and then look now with the single cell studies that Xavier and Salim are delivering to us. And we can see now for all these clusters, we see a cluster which is associated with a given function. So this is the cell cycle, this is the estrogen receptor, this is epithelial mesenchymal transition, this is the immune cluster. And when we look at because here in the rows are the genes, in the columns are the CPGs. And actually the p-values of associations are plotted here. If we look at those transcriptionally active molecules for instance, in the immune cluster, we see in the single cell data that indeed they come from the immune cells. This plot says this, right? And if we look at the EMTs, we see that they come from fibroblasts. So this in a way shows us also how you can actually use methylation data to ascertain at least the cell types or the tissues from which it can come. So we have a similar line of research on microRNAs, which maybe in another time we can present in more detail. MicroRNAs are very small molecules that do control the expression, not like through methylation, but by slowing down or blocking the translation, the production of the protein. And Miriam Aure and Anthony Matelier, other senior scientists, have been dedicated in my group, Anthony has his own group at the NCMM, but Miriam has been in my group, dedicated to study the crosstalk between microRNAs and, on one hand, methylation, but also on the other hand, DNA methylation. And this was something that I was intrigued when you asked Thomas, what comes first? One of these events comes first. So one interesting thing that I want to highlight here. So here again, like in the methylation, we see the same thing. First of all, if we do plot the p-values of correlation between microRNA and mRNA, we see 3 clusters. And these 3 clusters, again, guess what? They again recapitulate the same cell types. They recapitulate the lymphocytes, the fibroblasts, and the estrogen receptor signaling. So this tells us that especially in the estrogen receptor positive cancers, where we know what the signaling is and where there is a strict cascade of transcriptional regulation, we can recapitulate this no matter what we do, whether we do microRNA or methylation or expression. But what we found was a little bit, and we can also, again, in the same way, recapitulate this with single cell data. We say by also long non-coding RNA, yet another, not microRNAs, but long non-coding RNA, that we can see these 3 clusters. And we can also see that in these clusters, indeed, the expression comes from these types of cells, which we predict correctly. But in the case of the microRNAs, there was something interesting, that one of the microRNAs, 29, I'm not sure if I have a slide about this here, but 29C is a microRNA which directly targets the DNA methylase. So maybe at the very beginning, there is some selection in which MIR29 gets upregulated. And this may in itself be at least one of the reasons for activation of the methylases, which can be one of many mechanisms as to why we see this cascade of increasing methylation in the cancers, hypermethylation in the cancers. So one thing that one might want to say is that in the recent time, now I was presenting to you the results from our compendium of 2,000 consecutively collected breast cancers in the Oslo region, the way I described at the beginning. But most recently, we have been joining forces with clinical collaborators to do the same, but in the context of a clinical trial. So one such trial is this so-called NeoAva clinical trial that I want to describe for you very shortly. So in this case, these patients are both estrogen receptor positive and negative, but they have locally advanced cancers. They're very important that they're not HER2 negative, because if they're HER2 negative, they will get HER2 treatment immediately. So 150 patients have been included in this with locally advanced cancers. And because they have locally advanced cancers, they needed to have some neoadjuvant treatment to shrink the cancer to a size which is operable. And in this neoadjuvant setting, these patients, half of them got a classical chemotherapy, 4 cycles of FAC, plus followed. So this is a treatment that's supposed to kill dividing cells, followed by 12 weeks with taxanes, a treatment which is supposed to be a cytostatic to kill any tumor cells. In another arm, the patients were given the same treatment, but in addition to that, they were given bevacizumab, which is a treatment which is a anti-angiogenetic treatment. It is supposed to stop the formation of blood vessels. And at each time point, in the before treatment, at week 12 and week 25, we took core needle biopsy from that patient. And everything we found at each of those levels, at DNA, by sequencing the mutations, studying the copy number alterations, the mRNA, the microRNA, and the DNA methylation, is of course a topic of a different talk, right? But what we are trying to do in this is to both collect all these data at all these levels, look at all the interactions between them, the way I showed to you, how are things interrelated because of the central dogma or because they are connected in some form of regulatory networks, and identify these regulatory networks and show which pathways in which patient are responsible for the observed phenotype. Indeed, if you look at that, so if I told you that we can do copy number alterations on a tumor and DNA methylation and microRNA and mRNA, and you can ask the question, if I take any of these levels and I want to classify my patients by any of them, do I get the same? So this I did myself. I did this in Excel just to get an idea of that. So for everyone, you see that you get classification. And for every level, you see that this classification gives some meaningful results in terms of survival. So you see a survival signature. But in some cases where the colored line stretches all the way, they all come into the same group. So for those ones here and here and here, if you have classified them by one method, you have classified them by any. But for others, it is not the case. So is this because I'm lacking power? This is 120 patients. I mean, if I would extend this to 10,000 patients, would then the number of classified the same way grow faster than the number of those that do not classify the same way, right? That's a question. And I believe not. I believe that actually there is some information that we are missing in this so-called misclassification. So how can we take advantage of this misclassification? Well, we can develop other methods in which we're not going to do what we did until now. What we do until now, we take all the genome and we take everything which looks alike and try to combine patients according to the things that look alike. But in this case, we may combine not patients, but pathways. Let's take all the pathways we know something about, like P53, for instance, cancer, P53, the most central pathway. And let's look in this pathway, if we can look at all the members in this pathway and then look each individual member, how is it affected at the DNA, the mRNA, the protein, or any metabolic level. So you understand? Here we actually turn the argument a little bit around. We now work with pathways and we look, is my pathway normal or is my pathway deviating in any member of this pathway? And like this, we can generate new data. This time we are not doing heat maps of expression, we are doing a heat map of pathways. So here we are asking the question, which are actually the pathways we see most deregulated in breast cancers? And this was early in the 2010, where this turned us into immunologists, because we saw that some of the we didn't find the estrogen receptor, as you would expect from what I told you at the beginning, that estrogen receptor is the most important factor in breast cancer. We found the immune cells, we found interleukins, we found endothelins, factors that are important for the differentiation of the endothelial and other factors. And if we look at how this is in the normal, so now you can see all these pathways in the normal, they're kind of very close to each other. In normal with high mammographic density, which we know is a factor for risk factor, we see quite similar, but maybe a little bit stronger patterns. In ductal carcinoma in situ, which I told you was the one that stays in the duct, we already see fully blown patterns of carcinoma. And so basically we could use our normal breast tissue as a reference to which pathways are indeed deregulated in the invasive carcinomas. And this we can do also on a single patient basis, and this becomes now very important in terms of the personalized therapy, because we can use this tool to say in which tumor, which pathway is particularly deregulated. And I will finish by then saying that this is also useful in terms, so I will finish more or less where also Salim finished. What are these data useful for? Of course, one thing that I already said on my slide number 2 or 3 is that we want from all this huge data create those barcodes or create those factors that really have something to say about the patient. Yes, but how do we find those factors? Of course, there are many statistical methods, which some of them showed here, but still, there is no clinical trial on a worldwide basis that has statistical power to nominate a molecular signature, unfortunately. If you have a trial of 200 patients, and if you have data dimension of the one I said, you always have type 1 error. one thing we could do, and that is how we joined forces with the department from which Salim comes and with whom we are best friends, the Department of Biostatistics, to actually deliver all these data to them. And this is what brought us to our own EU project. The idea of that EU project was, if we take all these molecular characteristics that we have, which are so difficult to get into our heads, they can get into the computers of these biostatisticians. And we can create in silico tumors. Now, these tumors are not going to be in a tissue. They're not going to be in a patient. They're going to be in silico. Every tumor will have these characteristics at all these levels. So can we create such avatars, molecular avatars, because this would have a great advantage. We can treat them as many times as we want, and we can start all over again, which we cannot do in a patient, right? In a patient, a decision is made, and then one needs to follow this treatment. And this is, in a way, in a circular form shown here. So in a trial like the one I showed with FEC and bevacizumab, we have all these measurements at the different weeks. These measurements are in terms of histology, they're in terms of dynamic images, MRI. We have a PhD student who is exactly working on the MRI. And to these, we also add molecular characteristics. These molecular characteristics can be how many blood vessels we have in the tissue. They can be how fast the cell cycle can be. They can be, this is exactly the improvisation. And in fact, if you go out in the literature, there is a lot of literature of different biological processes described in differential equations. So if you speak to my collaborator, who's the leader of this group, when he sees that, he says, this may teach me biology, because he wouldn't understand the text, but he would understand the differential equations. So of course, those equations are all prisoners of their biological system, right? I mean, we know the cell cycle from that particular cell line in which the parameters were measured. Right. Now we are trying to derive those parameters from our own data. We can have our own proliferation signature for every tumor, and we can know how quickly that tumor proliferates, at least compared to the other tumors in our data set. So we're trying to use this, and the benefit would be double. For those guys, we deliver the data so that they can create their models. But for us, we maybe opportunistically can use their models to see which of everything in our data is in fact useful to predict treatment response or to predict something useful for a clinician. And indeed, the first simulations show that one can identify different regimens of treatment in which tumor cells stop growing, or we can both mimic the treatment of the tumor as we see it, but also we can predict different ways which might have ended up in a different outcome, so that we don't have that many tumor cells. And this is a little bit, so this one of the senior scientists in this, Alvaro Conluque, he compares this to prediction of hurricanes, those who follow the hurricanes in the Caribbean. Every now and then there is a hurricane. And then from simple basic parameters like the speed of the wind at a given time, the humidity, the direction of this and that, one can actually tell whether it will go to the right or to the left and with what speed. And every next time one does it, the prediction becomes more and more correct. So this is how we are trying to implement this into these mechanistic mathematical models, to which also Salim touched a little bit. And then with this, I will thank you. I'll tell you, this is my group. This is Thomas, who presented for us. This is Xavier, who couldn't present, but I really hope he'll come back. He has a great set of data and himself building his own group. Here we are in the mountains in Norway. It's called Nurifjell. And in fact, I'm just, the moment I stop finishing speaking here, I'm going there. In the that's place. It's 1.5 hours out of Oslo, up in the mountains. And in the summer, we get together in those, we have islands in front of the city. So it happens every time after the summer vacation, we celebrate that we are together with a picnic there. And I hope that one day you'll be able to join us either to the one or the other of those events.
Thank you so much. Thank you very much, Vesela. Good guns. And
We have guns too. We have guns too.
Okay, so you build guns. No, it's very interesting. First of all, specifically for me, was interesting that there are infiltration in some subset of patients with immune cells. I didn't know this, but what I knew from practical medicine, that allogeneic transplantation. By the way, Salim mentioned just transplantation, stem cell transplantation, as a tool which let us give stronger chemotherapy, but in fact allogeneic transplantation is a very strong immune tool, because this graft versus tumor effect is the major goal, especially in hemo-resistant malignancies. But in some cases it works, like in some leukemias, or in some of them yes, in some of them no, some lymphomas. But in many solid tumors, it doesn't work for some reasons. So probably there in between subset, but it works. Some evidence, it can be statistically insignificant in small groups, single center studies, but there's some patients response, which is not a scientific sentence, but you see these small signatures, the green dots as you show this, and maybe I don't know, this can be helpful in real selection of subdivision of standard at present standard classification of patients for HER2 positive, ER positive, I don't know, whatever. And there can be different approaches, because transplantation is quite aggressive and too many side effects, and maybe if patient has relatively good prognosis, it can be too much for the patient, because because chronic graft versus host disease is a terrible condition. Sometimes so a couple of times committed suicide, because these conditions can bring to terrible situation with patient, irreversible in fact.
Yes. So I agree with
But
you that our methods are quite applicable to these situations as well. So
maybe this is number one, to select patients who would have a predictable response for this. And secondly, there are, different approaches, for example, with the chimeric antigen receptors for leukemia. So chimeric antigen against BTK, B-cell leukemia, or something else, I don't remember exactly, or was inserted in T cells and in cytotoxic T cells, and it gave very good clinical results, with very good response rate in otherwise absolutely resistant children with acute lymphoblastic leukemia.
yeah.
So this can be happens, because sometimes breast cancer is dramatic. I just not like a doctor, because I don't work with this patient, but I know very bad cases with bad development, and this can be another approach. I don't know. And of course, what is brilliant is this pathway approach. I think this is very, very, for me, it's absolutely new idea, because this number of molecules, but it's very difficult to estimate and evaluate. And if we have, I don't know, the barcode of pathways, it can be very extremely interesting. Those heroes who has no small children in children garden. If you have questions to Vesela, it's just a time to ask. Any questions? Okay, so we have some participants left for last several minutes, I think, and it's easy to explain. But I want to say from myself and from all our audience, I think, thank you. It was very, very useful for me personally, and I think for those who participated in these 2 days seminars, because a lot of information, and I think that maybe this my fault, that I did not push and involve more clinicians, people from clinical departments to participate in this, because understanding of basics and understanding not only conclusions from the laboratory, this is increased, this is decreased, what happens there, what's under this, what behind this, it's very important. What is the sensitivity of method, what we are looking for when we give you positive or negative result. It's very important in new level of understanding things. So in next session, maybe, yes, Anton, I will put more attention for clinical department, especially there are many young people there, and I think it would be great if they will participate with us in the next session of seminars. Thank you. And Anton, if you want to add something and conclude as a president.
Well, I'd like to thank Vesela and her team for responding so quickly to the sudden changes in the condition that happened. But it was really very exciting to listen to you, and I think we can have some questions in the follow-up in the next days, as people will watch the videos of this seminar.
Absolutely.
Also, there are people on the stream in YouTube who did not have a link here to speak directly, but they were watching. So we can come back to you with these questions. And
Yes.
yeah, we really hope that Igor will be able to visit and to discuss future projects together, and that we could have on-site seminar next year.
Exactly. We look forward to that.
So thanks
Indeed.
again. I also would like to mention at the end our sponsors with who support this is happening. The program European Research Networks of the Bulgarian Ministry of Education and Science and the European Commission Transplant project. Thanks to you everybody for participating. Take care, be healthy, and see you in person, hopefully.
Yes, yes, yes. We stay in touch, Igor, about your visit, and maybe we can start planning some experiments. I saw that Manilo was joining us as well. Maybe we could have some
now we
discussions further
prepared
on how to.
time. So, and of course, I want to add. I had a poor connection, and I want to add the good words about your presenters, Salim and sorry, I forgot Thomas, right?
Thomas and Tom.
So this, and Tom, of course, yes. But today I mentioned those who participated today. And Tom was Tom, of course. Tom, of course. Yes, it was excellent. No words about this. So all docents, all lecturers were perfect.
Thank you. Thank you. Thank you. Thank you all for listening. So
this is very, very
Bye-bye.
important.
bye-bye, Anton.
Bye.